Kia koutou katoa, nā mai, haere mai. Welcome everybody. I'm Michael Kingsley. I'm the Interim Dean of Science at Waipapa Taumatarau, the University of Auckland. It's my pleasure to welcome you to the inaugural lecture of Professor Klaus Lehnert, a scientist whose work delves into the intricate machinery of life at the molecular level. Klaus is a functional biologist driven by the question, how do living things work? His journey began in Germany, where a childhood curiosity of freshwater vertebrates evolved into a career in molecular genetics. He earned his graduate degree in biology from Darmstadt University and completed his master's and PhD at Roche Molecular Diagnostics. After what he describes as the dream postdoc project right here at the university, at the Faculty of Medical and Health Sciences, he transitioned to leading a genome research program at Vilactia Biosciences. Today, he's at the forefront of using emerging technologies like genome sequencing to uncover the causes of rare diseases in New Zealand families. His work is a collaborative effort, working closely with colleagues from the School of Biological Sciences and the Faculty of Medical and Health Sciences to answer key questions such as what went wrong and how does it work? Please join me in welcoming Professor Klaus Lehnert to the stand.
Thank you very much for coming. I'm massively overwhelmed by your kindness in attending and massively scared at the same time. After coming up with an abstract and finally making slides at the very last minute, I realized this is less a talk about technology, but a talk about people and essentially fellow travelers in the pathway of science. But I'm a techno geek. I'm not ashamed. And so I try to put in a few technologies that really enabled the research I was fortunate to be part of over the last half millennium or something like this, feels like. And if I make in time, I'm really keen on an outlook. Because for me, stuff we did in the past is stuff we did in the past. The future matters, far more interesting than the past. So I didn't plan on giving you a lecture on science. There's one or two science stories in there that I'm quite keen to convey, but mostly it's stories about science and scientists and how, at least I, came to have a very meandering journey, if I can call it that way. And that's really that. All right, diving right in. I think I'll probably use the keyboard. I was born in the southwestern corner of Germany, where the red dot is. And that's quite a peculiar corner, because it has coal. Lots of other regions in Germany has coal, but this one is in the border to France and Luxembourg. And in sort of a reasonably recent history, before World War II, the borders changed about 8 times. And the place where I was born and where my parental side of my family grew up was variously French or German at various times. And then I think as a consequence of World War I, this region came under a League of Nations mandate, was administered by France until 1935. So during the bad events of 1933, this region was French, not part of Germany, which resulted in a lot of people that had to flee. The rest of Germany fleeing to this region, and then obviously after 1935 having to move on. And after World War II, it remained French. France wanted to keep it, if I can put it this way. Good on France. So it actually joined the Western Germany only in 1954. So in other words, the history of my father and my mother is mostly as French nationals. There's reasonably ambiguity about on which side of the border I was actually born. And unfortunately, I don't know, because I don't know whether it was jokes by my parents or facts or not. But the other thing that results in people from that area don't really have strong sense of nationality. And I think, even though I was born well after these events, this is part of me. I really don't care about political and similar borders. If there's a mountain range, that matters. But somebody drawing a line on a map, not really. So, because the area had coal, the area attracted heavy industry, steel, which in a war situation attracts bombing raids, which doesn't always go well, doesn't hit the target, but it hits the forests. And the forests around the area I grew up as a little boy were full of water holes. By the time they were sort of 20 plus years old, they had filled and they had actually become freshwater habitats. And they were very safe, and this is an actual photo from that area. And they were reasonably safe habitats because they weren't very deep, especially in summer when you dared to go out there, so you couldn't really drown in there. They would dry out every now and then, so fish couldn't survive, which means no fish disease. And you could catch all kinds of critters in there, which I did. And that's why my first exposure to anything biological was the 50 frogs I brought home one day when I was about 5 years old. And 49 and a half of them escaped. And most of them did their thing through multiple sequential nights. And nobody could sleep in the house. Hang on. I should move this mouse away. That's my dad on the left, and then in the middle, and my dad who was born there, and all we know is his parents were born in this region as well. My granddad and my grandma, obviously. My dad was really at heart an artist. But at the very bottom left there is a famous painting by a painter called Karl called The Poor Poet. Some guy who sits in a cramped room under the roof with a leaking ceiling, dedicated to poetry. And in great retrospect, I think this was my dad's life plan, which isn't too different from being a scientist. And he worked as a house painter, but he went to art school, studied fine arts, everything from painting to sculpture and all these kinds of things, and he really enjoyed this. I don't think any other picture portrays my dad's spirit more than the one on top in the middle. But then, no, I came along. Realities come in, and my dad decided to study architecture. And he became an architect and worked as an architect for the rest of his life. An architect sort of in the artistic sense of the word. Designing and building houses, including the interior. Very much like it was the School of Architecture in the 60s and 50s and 60s. And some years ago, Roswell and I went to Copenhagen on some business trip, and we ended up in a hotel that was originally left with furniture from the 1950s, the Scandinavian School. And oh my God, he would have loved it. He would still be there. We wouldn't have dragged him away. On the right-hand side is my mom. And we don't know the origin of mom. Mom lost her parents very early in the war. And I don't know whether she had a recollection of her origins. She only once told me, and it wasn't spoken of, and she only once told me that she was from Eastern Germany as one of the millions of displaced people after World War II. So I still do not know, and maybe once I retire, I'll do my X chromosome and figure out where my X chromosome comes from in the world. But maybe not. And the bottom is my favorite picture of my mom with our first daughter, which she had the pleasure to enjoy. So I must have. So I grew up quite all right. Had lots of fun. I had blonde locks, so I think I was destined for a surfer career. Not that there's any waves down there in this region. In the middle, I'm in my trusty speedy car. That was actually a Bugatti. It was full metal, and you can't tell, but it has been to the panel beater many times to be fixed again. And according to my grandma, the picture in the bottom right is the proverbial Klaus. Always guilty of something, and best not ask what happened. But I grew up quite happily and roamed around there and had pretty much free reign. Something that would scare the living daylight out of me if my daughters did this when they were 5 or so. But I must have been the perfect child because my parents wanted more like me. Or in the words of my sister, they tried to improve on version 1. And after a while, there were 4 of us. And this is a photo from, I think, the 70s, must be the 70s. It hung in our living room as a big poster for a long time, so it got a bit sun-bleached. And I just managed to bully my little brother into taking a photo with his camera and sending it to me for tonight. So you can't see that the car is orange, because in the 70s, even BMWs were orange. But this is quite typical. We went out into the woods and I went into the watering holes. So we moved to the family growing. Dad decided to build a house and moved to the Frankfurt region. And we moved into the house well before it was finished. I don't think it's finished yet. It's a project was always under progression. But I learned, so essentially we built it after we moved in the weekends and nights. So I learned everything from concrete pouring to gilding. And I'm quite happy that I acquired these practical skills, and if I ever get to use them again. And Frankfurt, center of chemical industry, what happens during wars, it attracts bombers do things, there were no watering holes. And I was very happy to go out and do more fossing around. But also there's reasonable transport infrastructure, autobahns. And when they build autobahns, the way they build them in Germany at least, they have those cloverleaf intersections. Really good. Wherever there's an exit, there's also an on-ramp in the opposite direction. Not as confusing as Auckland. Take that, LSE. And to build these overbridges, they dig holes. And that leaves holes in the ground, essentially clay pits, the moment after the diggers have done their thing. And because they were in the neighborhood, we drove past them, and you could see sort of these clay pits over 5-6 years turning into habitats. And I was very interested in figuring out what changes. And then later when I went to university, this is successive something, there's a term for it. But I would probably these days study this by just sequencing the living daylight out of everything in there. Also at university taught me that if I'm interested in this, I would be an ecologist, and that was a bit scary because I don't think ecology is understanding. The other very interesting part of the area is that it was surrounded. It was all over the place. Where US missile bases as part of the defense shield in the 80s, and US helicopter bases. And there was lots of troops. And which means some of the kids of the US troops actually went to school with us. So we grew up in school with quite a few American mates, which was great, made our English a lot better, even though they were very keen to integrate it by learning German. We very actively counteracted that. But also, once we became teens, this was first-hand access to the best, coolest, newest funk music ever, because the troops had access to this via the latest recordings in the PXs. So and Frankfurt probably being the most cosmopolitan place in Germany. I have a sense of being brought up in quite a cosmopolitan way, sort of reinforcing this. Who cares about boundaries? So anyway, these watering holes, I went out and caught and bred and fed all kinds of critters. These were my favorites, including a fish on the right, who is actually from the Amazon that can't find us in the watering holes, which feeds its young by secreting protein-rich mucus on the skin, which is actually very antibody rich. And is Steve here? Steve was trying to convince me that is the first foray into lactation biology. I'm not so sure. But this was actually hard work keeping them alive because you had to feed them, and in winter there's nothing you can catch to feed them. So it was actually grown Drosophila as a food source and was sourcing strains from universities just to breed them. And also breed all kinds of insect larvae, including the ones on the left. They were called bloodworm. I'm sure that's not their scientific name. And just as an example how much this affects my family, they don't survive for more than a day if you don't give them fresh water all the time. And that wasn't so keen for me to keep the tap running all the time. So I came up with the glorious idea of putting them into some kind of net and putting them into the cistern of the toilet. So every time somebody flushed, they get 9 liters of fresh water, which worked beautifully well until that fateful day when there were screams coming from the bathroom. And it just happened that my sister looked into it after doing whatever she did, and there were little red wigglers on the I just managed to stop them from running off to the doctors. And afterwards, I had great trouble growing them. But yeah, it was hard work keeping these things alive. I was ever out there scavenging fish tanks. Inorganic collection days were a highlight of the year, 4 times a year. Building fish tanks, building filters. The water around the place was quite mineral rich, so I had to turn this into distilled water all the time. So I built things like anion exchange, a lot of stuff to build because I can't afford to buy it anyway. And what fun is that? That's not my bike. My bike was a proper racing bike. And I had a bit of bigger fish tank on it. It's not my filter because mine wouldn't have leaked. No towels around it. And it's not my anion exchange. I don't have photos of those things because mine would be digitally controlled. So I learned by building, and I learned by taking things apart. Things while they still work. And to the day, I suggest whenever we buy some new piece of kit, while it's still working, that I take it apart to study the inside. Nobody's really keen on that. Somehow they think I can't put it back together, but no, warranty void, if steel broken, that's just an invitation. And just in case you're wondering whether that's all a bit nuts, I'm not Sheldon, so I can't say my mom had me tested. But the guy in the middle is Konrad Lorenz, who got a Nobel Prize for animal behavior and was sort of the founder of modern animal behavior, and I think in 1973. He got, I think, $300,000 for his Nobel Prize, and he used to build himself a freaking big aquarium. So I thought that's a quite nice way to fund your aquarium hobby. Just get a Nobel. Still working on that, of course. So as I said, this was hard work. It took a lot of time, which meant I didn't have a lot of time for school. And I'm not sure what to say about my schooling years. I don't think I was a bad student. I think 97% of my teachers would disagree. But mostly I didn't have the time to go there and to do things because other things interesting. So the one story I want to share is with my best mate Matthias, who now lives in Norway and tries to get the Greens into parliament. It's working even less well than here. Probably has to change his facial expression for the posters. And the teacher one day came up with asking us what plants can do to stop being eaten by caterpillars. And to the teacher's credit, this was in the context of plant wall thickness. Joe, you know much more about this than me. And fair enough, that may have been the right answer, but we massively disagreed because we had a much better idea. If we were a plant, we'd stop the caterpillars by secreting something that makes them form chrysalises earlier than otherwise. Because then they stop gnawing away, they turn into butterflies and bugger off. Anyway, because I was quite generous with my suggestions for improvement to the teachers, which invariably resulted in a very unhealthy desire by the teachers to meet my parents very early in term. My parents were very young when we had me, so mostly I won those things. For the teachers to suggest we enter in this German competition of some youth science competition. It's called Youth Researchers, if it's translated directly. But it means that if you enroll, the school has to make resources available. Funding. And at the time, we were going through the neighborhood in the middle of the night, stealing twigs and branches, extracting them with all kinds of alcoholic solvents, and shooting them up the rear end of caterpillars, which stopped them gnawing on plants immediately, mostly because they were leaking, rather lethally. So the solution was, we need a syringe with a much, much finer needle. All right, school, buy us 2 Hamilton syringes. And I swear the needles were much, much finer than the one in the picture, because we broke them immediately. But the school was good enough to find us replacements. And so we did this for a while, shot them up, and lo and behold, the chrysalises of this kind of butterfly, the caterpillars of this kind of butterfly formed chrysalises 3 days earlier. And we injected them with an extract from, I think it's English yew. It's an ornamental plant in continental Europe. So that was real fun, because we found out something that we didn't know beforehand. The teachers didn't know anything about it either. I have no idea whether it was known in the literature. I had a quick scan beforehand, before this lecture. There's one paper. I didn't list this species. But that was real fun, because we could do it. It was nothing to learn from books. Created lots of problems for the school, of course. The other thing that teacher told us is that if you do really, so I was still struggling with time in 2 ways. Finding time to do anything at school because of all the other things, and also 3 years to whatever NCA equivalent here. God, how long. So the teacher told me that if you do really well, and I think it was year, 2nd semester of year 12, you can request an early NCA. You can force the school to run NCA for you half a year earlier. Never, I think, has the world witnessed the improvement in grades in that short time, just because there was a chance to get out early. The school, of course, struggled to get the examination together in the subjects I wanted. So they didn't allow me to do physics. No, genetics and chemistry was a given. So I had to come up with some other things. I like to study failures. So I was not interested much in the politics of the Weimar Republic. This is the period between World War I and II, before the Nazis did what they did. Essentially a falling apart of a government system because it became too fractious. So I came up with this term, with this topic of the failure of the Weimar Republic and reasons of it. That this was apparently social studies, and then they wanted to be really bad to me and said, "Oh, you need to have an art subject as well." Well, too bad for you guys. Because of all the exposure through my dad, I'm really, really interested in art, so I chose pop art. And there may have been fashion design in there as well, but it was the 80s. Let's not talk about the fashion of the 80s. But there were models involved. But I had to talk about this pop art picture by Roy Lichtenstein for 2 hours. So if I run over time today, you get the better end of the stick. So I was released early from school for really, really, really bad behavior. And at the time, the thing next to this is military service. And you could choose to do this before or after university. And I thought, oh, well, I'm out early half a year. If I get into military service really quickly, I can get this over real quickly as well. It was not optional. You had to go to courts, multiple rounds to actually get out. So if you join the army, lots of walking. I don't like walking. That's why I had a bike. In the Air Force, no conscripts or draftees ever get anywhere near an airplane. Why? And I like anything to do with boats, so I volunteered for the Navy. Nobody from my region, central Germany, 700 kilometers away from anything salty, volunteered for the Navy, so it was easy. And I went there and yada yada training. And the prize posting was this ship. Is a 3-mast bark or 3-mast bark. That's an officer training ship. They still use it today. It doesn't have a wooden hull. It's deceiving. And so Well, much rather be on a boat with sails than a boat with diesel engine, I thought. So was working quite hard to get onto that. And then on day one, I found out that the job for people with my specialization was to polish the brass ship's clock once a day, every day. That wasn't so interesting. So I did the unthinkable and asked for a transfer to a destroyer fleet, which was part of the Standing Naval Force Atlantic at the time, because, 80s, and tension, and arms race, and all these kinds of things, which was great because it was a fleet of the NATO nations. And so you could actually hop between ships quite easily. You just had to let the next commander or CO know that, I'm free the next 6 weeks because my ship's in port, and I'm happy to come along. So I could hop between American, British, French, Spanish ships, and I spent a lot of time at sea because it's so boring when you're on land and you have to do cleaning all day long. and I learned a lot of really interesting stuff, mostly navigation, communication, and believe it or not, they were very keen for me to take things apart like transmitters while they were still working. After I made this argument, well, in a battle situation, I need to know, I won't have time to do it. So I got to take apart really, really expensive pieces of kit. But then every ship had 3 of them, just in case, so if I broke one. But I knew what I wanted to do thereabouts afterwards, and even then, long nights on shift. So I brought myself a few books along. There were 2 or 3 others, which I can't remember, but it was those. A German book on classical genetics, Drosophila crosses. I think most of you know Benjamin Lewis gene. I think it was a very early edition. And somehow I bought an old copy of Lehnert biochemistry. That thick, very, very thin pages, and hardbound. And that's how I learned that in a mixed environment, people who want to learn are not always welcome, especially when there's drinks involved. So I got beaten up with the hardbound copy of Lehnert 3 times, just because. But anyway, it just happened that at the end, we were counting down the days to release, freedom, after the service was over. And it just happened that we were in some kind of fleet training center in the Caribbean, in the biggest American naval training facility they have in the Caribbean, on the biggest Caribbean island, which I'm not allowed to mention probably. And it happened that, I think it was a Dutch ship that was supposed to be doing training exercise with us, didn't come in time. So this meant we couldn't go home in time for me to be in Germany on the day of my release. They started this, all right, we'll release you on the due date, but we immediately draft you back as a reservist, so you can be on the ship when we go home. To which I said, well, but as a reservist, I'll be an officer. So for 30 days, I was a Marine officer. And unfortunately, at sea, you don't wear a white uniform, officer and gentleman. So I didn't get the maximum benefit from it. Definitely not with the ladies. So I was actually released late for doing really, really well. So very, very mixed signals about performance. Would I do it again? No, not voluntarily, but oh my God, you learn a lot. Internet, 11 bits per second. War communications. So then because of, in Germany, university access entrance is a bit different from here. You can't apply to universities. There's a central, organization that awards you a place to study in a subject of your application at a university of their choosing. And this is all done by grade. Because I did reasonably well in my final exam and did it early, there was a deduction on the grade. And because I had my military service done already, there was another deduction on my grade. So I had a mathematically impossibly low grade, which means I could choose whatever and wherever. And everybody wanted to do medicine. I didn't really think so. And in the end of the day, I chose a small university, sort of 60K from home, because I don't like big universities. Big by me, bigger than this one. And close to home meant I didn't have to flatten, didn't have to find money and this kind of thing. But no, studying was free, and probably still is free in Germany. So that was Darmstadt, which is just south of Frankfurt. But I was really, really disappointed. I was, for probably 2 years, very close to drop out, because we mostly learned how to figure out what the name of this beetle is by counting appendices on the leg. Apologies to all the... Hopefully Greg isn't here today yet. And the same with plants. And in the end, after 2 years of memorizing and learning, all I learned to do is put a name to a thing. I still didn't know what it did or what it couldn't do. So that was real tough. But there was this one thing that wasn't taught in the first 2 years, the undergrad years, that I only heard in one guest lecture. That was a yeast geneticist. Funny enough, he gave a lecture on human genetics. The kind of thing you would do, Russell, or you would have done at the time, was massively misleading. But I don't think it was intentional. Because I actually quite like the yeast thingy. Because what you see here is 4 spores out of baker's yeast. And they're the direct outcomes of a meiosis. So you can track meiosis one-to-one back to the sister chromatids. And that's very, very, very useful for mapping and crossing and all these kinds of things. Anyway, not sure I should mention this here. German professors are very, very powerful. They are government, federal employees for life. Try to get rid of them. Good luck. Students don't pay fees. No direct financial pressure on student numbers. Frankly, mostly they don't care how many students are there or what they think. The genetics professor, I really, really love him, but he was tough. But he had very clear and unambiguous rules. And the rules were, if you go through this program path, the genetics course and lab course, 1 year, was compulsory, even if you didn't want to do genetics afterwards. To get into this, you have to sit an exam at the start of year 3. Mind you, this is the start of the semester. The first week, there was no meaningful genetics teaching in year 2. So you had to figure it out yourself. And only 16 people you had to pass, reach the pass mark, and the 16 with the best pass mark would be allowed to do the course. And we were about 150, 200 in my year group. And 1 or 2 might be invited into a master's thesis, which meant that the entry exam would be taken by newbies like me, first year round, and people that have done this, have tried this for the 5th, 6th year running, because they had everything else done. They couldn't graduate from that one paper, which means they couldn't graduate overall. So I thought, that is actually a bit serious. That's not sure I can do that. And also, for that summer break, I had another project lined up. There was something to be built that had nothing to do with biology. But I had the immense, inexplicable, unforeseeable, unpredictable luck of a couple of years, year and a half earlier, prior to running into Tina. Love of my life, even though she'll kill me for saying this. And she had the glorious idea of not trying to get me away from, not to take the distraction away, but take me away from the distraction. And suggested, why don't we just go away? And we rented also an old cottage in Brittany, in France, and drove there in one rush through the night, 1,500 kilometers, in one go, only petrol stops. And had the most marvelous time there, and I never, ever studied as hard as in my life ever again. As for this, and it worked. It worked beautifully. So that meant I was assured a place in this research group for my master's. Because once you did do well in this exam, the lab course was actually quite straightforward. Well, one leads to the other, as you know. And I think it was 6 days before I was due to start. Yeah, it was 6 days before I was due to start to take up my lab after my final exam. I went in to do some preparative talk. And that genetics professor, laughing dearly, said, I've been asked to recommend somebody for a position. It's a great opportunity. You should go and check it out. And that was in southern Bavaria. Very good. Are you from there? Cool, all right. Hang on, let me modify my speech. And I went there, and yes, he was right. It was a tremendous opportunity. This was a town called Penzberg, not doesn't matter so much, but it was yet another coal town where coal mining had been given up, I think in the 50s. And unlike all the other towns, had massive federal and state government support to set up the equivalent of German Biotech Valley. So all the way from Munich to that place, which is about 100 kilometers, today is littered with established pharma, startup, Max Planck Institute, all kinds of sizes. And this was sort of building at that time, which was the great time of recombinant DNA and cloning and all these kinds of things. So yeah, there was no way that this was a bad idea, but it was also still yeast. And that's not it. They built a factory or the research facility on the old coal mining land. It was called Boehringer Mannheim at the time. It's called Roche Molecular Diagnostics today. And over 5 years, this completely turned the company around to the point that Roche came in and bought it outright. And for 4 years, I was part of that. And it is a fantastic feeling of being part of something like that. It didn't matter that money was no object. Sometimes it was, but just success after success after success. It's really something that if you ever have a chance to join something like this, do it at whatever cost. And the cost was high. It was 458 kilometers away from Tina. And Tina drove this weekend after weekend. We rode off an entire car. And there's a Google reckons it's 4 hours, 39 minutes, and that was during the sailing daytime, so middle of the night in Germany, no traffic. It's more like 5.5 hours. We never managed, we never got over 4 hours, unless there were traffic jams. It's a major miracle we survived. And the other thing was there was a fantastic peer group, that's them. People who'd done completely different things from me, but you were with friends, with mates, and all of them stayed with Roche. But my, they do winter really well in southern Bavaria. That wasn't so compatible with me. So that was my PhD, my master's and PhD. I won't talk about the master's, but I want to share something about PhD, and that's the first science story I want to tell. So in all organisms that have a nucleus, you, me, and baker's yeast, but not microbes, proteins need to get to the outside of the cell for all kinds of physiological reasons. And the way they get to the outside of the cell is not by the cell just popping open. There is a very controlled, regulated pathway that channels proteins through membranes outside. It's called the secretion pathway. Now, when proteins go through that secretion pathway, some of them need to be decorated with carbohydrate structures, called glycosylation. Again, has all kinds of biological, physiological functions, and it's also related to disease. And the way this works, this protein enters the secretory pathway, and then a precursor carbohydrate is transferred onto the protein. It's this, can you see my cursor? Yep. It's this core oligonucleotide on the left. All these little hexagons just represent sugar monomers, sugar molecules, like glucose and so on. And so it doesn't really matter what they are. So that's the same in all organisms with a nucleus. You, me, baker's yeast. But from then on, things are very, very different. In mammals, there are very, very, very few additional carbohydrates added of very peculiar types of sugar. While in yeast, yeast just goes completely because yeast completely goes bonkers. Adds hundreds upon hundreds upon hundreds of carbohydrates, all mannose residues, just another sugar. And that was well known. And it would end up with structures like this on the bottom, and there's not enough room to prevent all the carbohydrates. Sometimes it's over 1,000 per chain and then multiple chains per, which of course is a problem when you want to make therapeutic or diagnostic proteins. Therapeutic proteins, these things are immunogenic. The liver receptors love nothing more than mannose residues, terminal mannose residues for clearance. So the things go out faster than they go in. And if you want to put them onto electronic sensors, this bulky stuff hinders electron flow. So the job was to make a yeast strain that doesn't do these things. And the way we did it was random mutagenesis. We exposed a yeast strain to a random mutagen, can't remember, I think may have been ENU, and well, at a level where we sort of statistically we're hoping for one hit per gene. For one gene to be hit per cell, so that we don't create too much damage. And then we fed the cells with radioactive mannose. And the underlying idea was that the cells would incorporate the radioactive mannose into their carbohydrates and radiate themselves to death, because these proteins would also go into the cell wall. And the cells that we randomly, uncontrollably destroyed the gene that transferred these residues onto the core oligosaccharide would survive because they no longer could incorporate radioactivity into the chain. And then we stored them for a couple of months, I think, -80, just to increase their radiation damage. Then we isolated the mutant colonies. The idea was that only those that no longer do this can grow. And then we mated them with other mutants that were, combined them with other mutants that were known to carry other mutations in the glycosylation pathway. Then you could sporulate, stuff you could do with yeast. You can easily combine two. Takes 3 days, not 9 months, like in humans, if you could do it. And then select the double, triple mutants, and then come along and cleave off the carbohydrate structures and analyze them with fancy equipment and try to find those that no longer can do this. We actually had to clone an enzyme, identify from a wild type bug, an enzyme that actually catalyzes the cleavage of. It's called PNGASF. I'm sure Sean knows about it. And so if you buy any of those, that's the enzyme we cloned and made ever since, just for that project. And then ran a very special kind of HPLC. This runs on 100 millimolar sodium hydroxide to get the pH to 13.5, so that all the carbohydrates become anionic and could be separated based on charge. Everything 100 millimolar sodium hydroxide means after about an hour in front of the HPLC, you can't grasp anything anymore because everything is sodium hydroxide slippery. But the company, Roche, had one of these machines, which were brand new, and I went there at night and used it. Found out at some point in time that this was the machine that Roche bought to prove to the FDA that the carbohydrate structures of the other therapeutic proteins were up to spec. So this was an FDA-certified clinical trial validation, and I messed with it every night. Anyway, ended up with about 600 of those structures, all kinds of different combinations, and went home for 3 weeks to figure out how they essentially make up a biosynthetic pathway. And all of those are intermediates and partials and all these kinds of things. So it was the biggest Sudoku ever. But unlike a Sudoku, we didn't actually know whether there was only one solution or multiple. But after 3 weeks, I had a solution. God, there was no computers for that. Long story short, turns out that we really knocked out a gene that we called NGD29, N-glycosylation defective mutant 29, that catalyzed the addition of that first carbohydrate, mannose residue, and all others. And then they get elongated by by enzymes that were part of the normal pathway. But if you could prevent the addition of that first one, no others would be added, which then meant we actually ended up with the core carbohydrate, and we could transform into yeast genes encoding the enzymes for the mammalian type glycosylases that transferred sialic acid and all these kinds of important residues to it. And that was that turned into strains for pharmaceutical production. Here's one example where we expressed glucose oxidase, which is in pretty much every blood glucose strip that you can lay your hands on. On the left-hand side is the glucose oxidase expressed in the wild-type strain. Now you can see very, very large, very bulky, very heterogeneous, and most of this is carbohydrate. And here's this expressed in the glycosylation defective strain. It's a single oligosaccharide, and this was put into the first generation of Roche's electronic glucose molecules. I don't know whether it's still there today, because they don't tell me these things. But that was the first use, and even though the things look slightly less sexy than today, it was quite nice to see a product that you helped build. Retail packaging. It's quite a strange thing, seeing a retail packaging of something you made. And I quite like this. And we did this with all kinds of other proteins for therapeutic purpose, GCSF, for example. I think this is actually GCSF, and you can see it's expected monomeric species. And you could express these things in yeast and make them for therapeutic use, because yeast is a generally recognized safe organism for protein production. Yeah. And we did lots of others. So I need to talk about enabling technologies. PCR is one of them. And I think most of you know about PCRs. For those that don't, you make tiny little bits of DNA, and then you can multiply DNA as much as you want. It's an exponential process, and suddenly we could get access to DNA as much as you like. All right, that was the end of it, and Roche had all kinds of plans. I had a Genentech postdoc lined up because they were very keen to send me to send their people to Genentech instead of just hiring all of them. But Tina and I had some other plans. We decided to take a break, and we traveled around a bit. And we traveled around the South Pacific, spent 4 or 5 weeks in New Zealand in July, August, in a motorhome, mostly in the South Island. We froze to death in a place called Twizel. And after a while, we got a bit fed up with the driving around, decided to find ourselves a nice place and stay. That nice place turned out to be Whitianga, and we decided to get married there. So that's the official wedding photo. There has to be boats in the wedding photo. And it was Tina, me, and the guy who ran the local Hammer hardware shop, who was the celebrant, and a nice lady who ran the local tourist office, Moira. 4 of us, exactly the way we wanted it. And everybody at home was rather grumpy. But by the time we arrived, we went to the Pacific Islands to warm up a bit. That was over. And then, long story short, I embarked on a postdoc thingy. So I read in one of those nights in front of this HPLC machine, Trends in Protein or Genetics, one of these, a review on cell adhesion molecules. Know nothing about them. And I quite like the fact that they do mechanical signal transduction. I thought, that could be an interesting thing. So I was trying to find a postdoc position that did anything remotely doing. Had already given up. Then, and one of the farewell presents from Roche, or presents after graduation, was a subscription to Nature paper. So every week, Nature issue would arrive. And at around 2 or so at night, I found this ad from Jeff Christensen in med school. Hey, cell adhesion molecules. In the most unlikely of places at med school up there. And I do remember waking Tina sometime around 3. Hey, Tina, would we go to Auckland? Hey, I'm not sure she was fully awake, but hey, I had a license to apply. And I applied, and never mind talking about immigration. Months later, I arrived. Jeff picked me up. It was July again. It was raining like hell. We were driving from the airport to the city, And he was going on and on about how he couldn't wash his car because there was a water restriction going on. It was raining. The car was at great risk of being washed off the water, not in Mandarin. And that's how I learned how Auckland manages water or not. But I arrived now, 25-hour flight, massively jet-lagged in the lab, with all this kind. I really didn't know where I was and whether I was in the same place. Actually, this should be the vision. But where's Euphemia? There was Euphemia. Euphemia was in the lab. I met Euphemia on day one. Bill as well, and I think Rohan on the same day or day after. Because Euphemia socialized me. One of the reasons why I'm still here is Euphemia. So I can't see you. Thank you very much. Thank you very much, Euphemia. Euphemia does everything via lunches. And we had lots of lunches. And so the project was to demonstrate that cell adhesion molecules can deliver the second sector to stimulate T cells to expand and do whatever T cells do. This was well known for other molecules, classical co-stimulator molecules like B7, and the postdoc project was to demonstrate this for cell adhesion molecules. And I don't have time to go through this, but those of you who are known to these figures, these show that 3 different cell adhesion molecules that we tested, VCAM, MADCAM, very aptly named MADCAM, and another one called ICAM, co-stimulate T cell proliferation as well or better or with more greater efficiency than B7. They amplify the B7 signal strongly, they induce hyper-responsiveness to it, and most excitingly, they could be delivered remote on a different surface. They didn't have to be on the same cell. We put them onto plastic beads and injected tiny plastic beads. We injected them into tumors, and the tumors went away with 2 weeks. Cherry-sized tumors on the size of little mice. But we gave the mice the tumors in the first instance, its model system. So the only way to move on with this is clinical trials, which is, of course, a difficult thing. So meanwhile, Chiara was born, our first daughter. And of course, she had to start life in a boat. What you can't see in the photo is that the paint is still fresh. It was finished the night before she came home. No, it wasn't finished. And we managed to acquire a restoration project in the north shore, far, far, far away. But I had help and an instructor. So we've been renovating this place ever since. And the other thing that happened along the same time was Jeff stitching a deal with a company called Human Genome Sciences, which was a company set up by Craig Venter at the tail end of the Human Genome Project, with the overall idea of making gazillions of money out of the Human Genome Project. But they had very, very, very large databases of partially sequenced RNA. And they allowed us to search them. So Jeff came home with this deal, and nobody knew how to search. And they didn't send us the databases, they were remote. So I've been pottering around with programming for a while, but there was never any real good use for it. So this was the birth of bioinformatics in my life. Taught myself bioinformatics, and this is me in the old office. Got myself a computer from a small, dingy outfit in Penmuir called PB Technologies. Single guy, single shop upstairs. Clearly he was in the right business. And later I met a guy called Dave Saul here at SBS who ran a Unix workstation. And he allowed me to hook up for it. And we then done this all. And in one night, while Jeff and the lab team was screening with filters for cell adhesion molecules for months and months and months, breathing all kinds of organic solvents, I found 2 new cell adhesion molecule genes just by searching databases. Oh well, that might be the way. So I wanted to get into this what then was called genomics. Then there was really the place to go was Genesis. They had similar problems, and can't mention that you want to go into genomics to people like Euphemia because next day you talk to Jim Watson. So I actually went to Genesis. It was the worst timing in my life. Nothing to do with Genesis, but literally in the first week Genesis ran into some financial trouble because I think some trial failed or something like this. And Russell rang. And Russell rang. I won't tell the Violectia story because the Violectia story, the start of Violectia is really, really Russell's story. And Russell should tell it. And Russell has never given an inaugural lecture. So here's the opportunity. Let's history of Museum biotech. And I was invited to an interview. I met Chris Ford before. Where's Chris? Hey, Chris. Met Chris Ford before because some equipment access in the med school. And this is the original document of our first meeting. It's fair to say that nobody really got a word in. Russell did. There was Russell, Chris. I think I took great comfort from the fact that Chris was there because Chris is a very, very nice person. Russell is too, but you never know beforehand. But the HR consultant had to have a bit of a light out afterwards. And if you look at it closely, we've done everything that is on this plan. This was the master plan. Over the lifetime of Violectia, we've done it all. Russell, very, very careful of his people, said, hang on, let's not start immediately because madness was going on in the country. The Royal Commission on Genetic Modification, minor celebrities were running PR. I spare you the more offensive posters that they put up and down. There was great risk there. Even the kinds of things that Violectia was intending to do, which had nothing to do with genetic modification in the bovine world, would not be allowed. So there was 3-4 months of just completely lost progress. But on the other hand, meanwhile, Jana was born and she also had to grow up in a boat. This time I thought I built a proper one. Didn't finish. But we put her in there anyway. It's still in that state, grandchildren. And again, enabling technologies, we had to build gene expression microarrays. Suzanne made a fantastic library of essentially all genes expressed in bovine tissue, and we sequenced the living daylight out of it. And the bioinformatics job was turned most to add gene symbols to it. Mind you, there was no bovine genome. There was just barely a human genome. So this was a massive sequencing bioinformatics project. John Pearson, who's now AP in Otago, made all the statistics work. Wouldn't have worked without JP. And we made expression arrays. And we commercialized them with Agilent. Agilent sold them. This is the retail packaging. I'm obsessed with retail packaging of one of the first kits. They're still selling them today. And microarray technology is really dead, but they're still selling them today to industry. And then the really, really, really cool project started. This is a gem. There is no greater privilege in a scientist's life than being entrusted with running this idea of Russell's. And the idea is actually quite straightforward. He says, after not having had it himself. And this is a distribution of a phenotype, milk fat content in the New Zealand Frisian population. And you'd call this a normal distribution. Absolutely. And the way the dairy industry did work and still does work, unless you disagree, Richard, is working on sort of slightly on the right-hand side of the median and trying to move the entire population as a population closer towards whatever I consider the better end. And it works. It's very robust, has practically zero risk, and it's actually what keeps this country running to a large degree. Now, Russell's idea was a different one. Why don't we take the superior performers and breed from them? And screened the entire database, something like 4 million animals for milk fat content, and found handfuls of cows. Here's one example. And she has, I think it was about 4 standard deviations below the mean in milk fat content. And with a distribution like this, 4 standard deviations is a lot of distance to the mean. A very unique cow. She made very little fat. And the little fat she made was highly unsaturated. Very healthy fat. And most likely because she didn't have to put so much energy into making milk, she made a lot more milk. direct profit for the farmers. And no, this is finding this animal is clearly the achievement of Richard Russell, a guy called Alistair McGibbon, who is the most accomplished scientist in the world in terms of milk lipids. I think this is a fair statement. Who said famous things like, if you could make butter from this cow, this butter would spread straight from the fridge and would be healthy like margarine. Here was a name for the cow. And Alex Angus McCurdy, most beautiful person on the planet, who can get farmers on board with practically everything. She ran all the animals for us. Nothing would have worked without Alex. Has to be said, and it's true. If you have an large animal try to run, go and contract Alex to run it for you. And of course, there were plenty of experts in the industry, including our own scientific advisory board, that said things like, that's just a bunch of genetic variations coming together, lots of them. You'll never keep them together. They segregate apart. It's environmental. Don't bother. So how do you prove this? Well, you have to make kids, offspring. And we made offspring. We made 7 daughters and 4 sons, actually 5 sons of them, via embryo transfer. And then it takes 3 years for a young cow to make kids, to make milk again. So 3 years of waiting. And this is a plot that makes me both very, very happy and makes me both very, very grumpy, because I should have started plotting the data 2 weeks earlier. Each data point is 1 week apart. And what's plotted here is milk fat content in 6 offspring from that cow. There were only 6 lactating at the time, and the right hand are controls. And the right-hand side panel, the solid line shows the milk fat content in typical Friesians, wild-type animals. And the dotted line shows the milk fat content that it was typical of mom. And kids, if you ever have to fake genetic segregation data, don't fake them like this, because it's just too bloody perfect. It's segregated 3 to 3. Perfect Mendelian. I think it was about around midnight when I emailed you, Russell. And I think we started next morning, 5 a.m., turning the science project around. But the next thing you can do actually requires more breeding, 3 more years. So generated lots of excitement in the characterists and the media in New Zealand. It wasn't so much fun when they rocked up on Saturday morning in my driveway with a camera and I had no authority to speak. Take that from Terra Communications. We did everything we could to find an interesting gene. Literally throw all the technology we had at it. Russ and I went on a pilgrimage to Paris to the patron saint of genes and made our offering. They wanted €1. We didn't have €1. We had only about 80 cents. We were punished, as you will see in a while. And also, meanwhile, Via Lectia moved out of the commercial diaspora, that some commercial wisdom decided that we have to get the academics out of academia, so they sniff some commercial air in some godforsaken place in Ellerslie. Commercial building where all you run into is call center people and corporates. And we came back as the anchor tenant in the IIB. That's another deal that Russell has to talk about, not me. And then 3 years later, we had more segregation and offspring. And it was not recessive. It did not segregate apart. It was freaking dominant, and it obeyed Mendelian rules abso-freaking-lutely. I shouldn't say abso-freaking-lutely. But it's a beautiful, high penetrance phenotype that segregates. And from then on, it was actually, we did a GBOS with only 111 animals. Sharon Browning did it, because we thought it would require special skills. And then it was quite easy to find the gene. We find the de novo mutation in the most boring gene in the planet, because we knew all about it. The single nucleotide substitution, which changed, I would have been called a substitution from a methionine to a leucine, which everybody who knows about protein chemistry can make all kinds of causality conclusions, but no, this actually turned out to be an exonic splicing enhancer. It took out an entire exon. It was an in-phase exon, so the RNA survived. but the protein it encoded was enzymatically inactive. And we stuck it to baker's yeast to test the protein activity. And that was all Hamish's doing. This was a major achievement to get the assay going. Radioactive. And yeah, all credit to Hamish. So that was then around 2008, 2000. That was the bovine team at the time. I put up the slide because everyone on the slide deserves massive credit. This wasn't the only program in Via Lectia. Sarah and Steve, who had joined us by then, who actually knows everything about dairy physiology, had a major impact. Elena did much of the technical stuff, and Hamish. So all credit to those. Alex, obviously based in the Waikato. All credit belongs to this group. And looking back, I think Via Lectia really conquered dairy genetics in New Zealand, in bovine. We discovered every single genetic variant underlying, reasonably frequent and significant changes to whatever the phenotypic trait in milk is, and some health things. These are just example papers. There's a whole bunch of patents. We secured all of this for the New Zealand dairy industry in form of IP. And it wasn't just, definitely not me. This was a collaboration internationally and national. Well-known people in Henry in FHMS, Nicholas down at Messe, international people, including members of our scientific advisory board, who did a very, very, very good job of making our life hard. Mostly by disagreeing that this was a good idea. So it was a hard fight to keep this program running. And I think one or two of Russell's gray hairs are directly attributable to that. All right, meanwhile, new technology came along. At the end of this phase in the lecture, called massively parallel short-read sequencing, we could suddenly sequence genomes. And we sequenced genomes in Via Lectia, sequenced cow genomes, well before Illumina was on the market. We paid Illumina to do it for us, but we did all the bioinformatics for it. And it's a pretty straightforward process. And in 2009, there was this seminal paper where they took 12 individuals from 12 different families with the same condition, or looking like the same condition, and found different mutations in the same gene by just sequencing parts of the genome. And the message from this paper was, it can be done, but you need multiple individuals with the same condition that are unrelated. So we just solved, through slightly different ways, a single mutation in a single animal. And we were very interested in this for reasons I come to in a moment. So we thought, maybe this is what we should do. So leaving Via Lectia to set up something new, this is just geekery, lots of bioinformatics, lots of computing required, wouldn't have worked without the absolutely generous support of Mark Hagen, who at the time ran NESSI. Still today, we run the first University of Auckland NESSI project, UA0001. Over my dead body will we give up this number. And we were in the process of deploying this to something I'll talk about in a minute, when Rohan and Zitan rocked up. Rohan, who I've introduced in the first slide, I met in the first week, and they had a similar idea and wondered whether this could work for areas of their interest, common variable immunodeficiency disorders. And I remember a meeting with me and Rohan and lots of clinicians. I think, Bill, you were there too, in a small room at ADHP where lots of experts said it can't be done, it's environmental, it's not genetic, it's multigenic. Haven't we heard that before? And good on Rohan and Zitan for persevering. We sequenced 3 families and we found the causal mutation in 2 of the 3. Haven't given up on the 3rd one yet. And one of this was a mutation in NF-kappa B1, a very important transcription factor in immunology. And what you do when you do those things, you find lots of genetic variants and lots of genes. Any one of them could be. But the clue here was that mice carrying a mutation or knockout allele in NF-kappa B are nude. And some of the individuals in the pedigree had a tiny note in their clinical record that they had alopecia. So the clinical details matter. You can't just sequence and not know about your patients. And the other one was a gene completely unknown to the condition called TCF3. which encodes another well-known transcription factor in biology, important for B-cell maturation, which helped prove that the most well-known gene causing immunodeficiencies is not TEC1, but it's other genes. And this led Rohan to formulate a hypothesis of epistasis in CVID. So the reason we wanted to set this up was Jesse coming back from the US, and setting up this for neurodevelopmental conditions. So we set up a collaboration called Minds for Minds. Russell, Jesse, major players, Julia Taylor, our clinical geneticist, and lots of other researchers in there, which I can't fit on the slide. And that's sort of, and we sequenced many families over the years, and I put up these title snippets not to show that we have lots of publications. We can't keep up with publishing these things, but every single publication here represents one family, one New Zealand family who now know why they have the neurodevelopment condition. And for some of them, this has enabled clinical improvements, better therapies, and so on. Lots of students involved, and lots of clinical collaborators. Again, I don't have time to go through all the names. You'll recognize yourself. And Suzanne has just published, put together a paper that's sort of a 10-year retrospective of what this program has done. So we've also played around with New Zealand other dairy industry, goat industry, and I put this up because I need to introduce Andrew Jiang. This has very little to do with the goat industry, but that's quite a different dairy industry. We started with figuring out whether they are highly inbred or not. They're not. They're essentially flocks of individual herds that are not genetically highly similar, unlike bovine dairy. We applied for an MB grant. MB announced that there would only be one grant awarded in this category. We applied anyway, we got it. And that gave us money and license to go out, collect samples. And we built the largest genetic and sample repository of dairy goats, I think still in the world, definitely at the time. And here's Andrew sitting in Alex's horse float. We rented ourselves a little high-ace van, put a centrifuge in the book, and literally drove through the Waikato for about 6 weeks. Here's Andrew in the back of Alex's horse float, which was the lab, happily dealing with the milk samples. I walked past and asked him whether he still knows which one is which, and he said, go away. And we found, and we promised in the grant, 4% improvement in production worth, I don't know, something like 15 million or so, that product. We found 20% improvement. And when we found this thereabouts in year one. And Russ and I said together that, all right, the research program is to find some more and do some more research on that one. But if we do this and delay the rollout, the grant plan was to finish the research and then commercialize after the grant. Why don't we try to roll it out now? Because 2 years, the farmers can make money. If you're in business, you can make the same amount of money now compared to 2 years earlier. It's worth a lot more now. That required change in the research contract. University wasn't so keen. I rang MBI anyway because I can't implicate Russell in that behavior. But we rang him and we talked to our contract manager. And yeah, of course, do what you want. Yeah, that sounds like a right idea. And then later, makes that much money. Hope you get a good cut of it. If the funding agencies think we should get a cut of it, maybe we should write this into grants. But and also another condition was, and whatever leftover money is there, just use it for research. So we used this to generate more genome sequences, which jump-started the following program, where we found another marker that makes goats 25% smaller at the right genotype, but still produce the same amount of milk. So that's production efficiency. So we actually commercialized it. We worked with egg research to make tests that farmers can send samples away now and get results back. Strange enough, we did not win the New Zealand Commercialization Award for that. So I'm getting to the end, guys. Meanwhile, always a meanwhile, new technology, single-cell transcriptomics. This means for single cells, individual single cells, we can find out which genes are active in those cells, which also means we can find out what they're capable of doing. Imagine 100,000 individual cells from some tissue or whatever. You get a full gene activity profile for every single cell. And you can find out which ones are similar. It's called clustering. And Andrew has done this in Huntington's disease from brain sections. And you can see the gene expression profiles are similar by cell type, similarity. And you can do all kinds of interesting things about function and infer modules and all these kinds of things. I don't have time to go into this now. It is absolutely clearly a transformative enabling technology. It's darn expensive, but so is rocket science. And Andrew, who was by then a PhD in this program, he's done a marvelous job. It's a stellar PhD thesis. He got off to Harvard, of course, to do more like this over there. And we also we applied the same technology in a fantastic collaboration with Rod, Sam, and Alicia, where we try to understand how cells communicate in tumor environments with tumors, how myeloid cells differentiate together, because you can order the cells by profile difference and infer what's called trajectories. And that's highly enjoyable. We need to find more money for that. So here's a list of funding agencies over the years. It was the Cancer Society funding that brought me to New Zealand. I forgot to mention that I came to New Zealand for a science project. I haven't seen much of the countryside since. All my colleagues that I showed you at Roche, they traveled New Zealand far more often than I have. And some other projects that are still ongoing that I'm involved in, they're highly enjoyable. Work with Bill on hepatitis B and liver cancer, Emma on motor neuron disease, Annika with and FHNS, founding mutation in Sweden. We had a lot of great collaboration with John Skinner, cardiac conduction, and we also figured out what gave a hippo at Hamilton epidermal bullosa. Something I wasn't quite sure how much I can mention is genomics for and of the New Zealand Neurological Foundation New Zealand Brain Bank, with supported by Richard Fall and Maurice. This is highly enabling. It will allow clinicians to stratify the samples from the brain bank, and we find causality mutations. This is actually the meanwhile slide. Meanwhile, when all this was going, the girls have graduated. Yeah, I graduated with a BSc in marine biology from this hello place, and now works with penguins and turtles. Looks like she has the time of her life. I'm very envious. Jana graduated with a BS, and up there with her partner Shako. Thanks for coming, Shako. Jana graduated with a BSc in social work. Works in child and adolescent mental health on the North Shore. Very, very tough job. So proud of you guys. And she's up there with her partner Sane, and they live their own lives. They do their thing, and Dad doesn't have to worry. Dad doesn't have an influence anymore, which is good. Not. So that is the end of the journey as such. But please allow me 3 outlooks, because, as I said, the next is always the more interesting thing. I see other transformative things on the horizon. AI, of course, but not AI in the dumb way. No, write my essay, summarize the paper for me. No. AI for new biological insights. And this comes from a PhD study of Irene that I co-supervised with Mark Hagen in ComSci, where Irene has retrained a published model that is very successful in predicting where transcript sequences, essentially genes, are spliced. There are sequences that are interrupting genes that are removed from the transcript to make make a molecule called spliced RNA that then can encode proteins, can be translated into proteins. And we know from, I don't know how many decades of research, that the mechanism, the biological mechanism, involves what's called an intermediate alariate, this bit, whereby this part of the intron is cleaved off and joined and covalently joined in a loop to an adenine residue in the intron. This is an obligatory intermediate step. This is then discarded, and this end is joined to this. Okay, that's just a biological fact. It's well known, and it's reasonably straightforward to identify those in about 80% of the cases. This adenine residue is unbelievably impossible to identify because these regions can be gigantically long, hundreds of thousands of nucleotides, of which, by definition, tens of thousands will be A's. There's no motive to identify them reliably. So what this model does, it predicts with 0.985 accuracy which of those two things are joined and which one are not. And when we went on to probe this model, can't lift the hood, can't look underneath, we probed it by mutating the known A's. There are only about 1,000 known A residues. The moment we change this A in the sequence to another one and ask it to predict, prediction accuracy drops to something like 20%. So to me, this means this model, which has been trained on nothing else but here's an exon, here's an intron, starts here, starts there, here's another exon. The model was not informed or trained with any information about the A, because we don't have it. This model has learned something about the mechanism. This model has learned that some kind of A, here, somewhere there, that we can't predict, identify, is critical for splicing. So what we want to do, what we should do as biologists, is adapt those models so that we can look under the hood. See what they have learned. Stuff that we haven't learned so far. This we kind of know. It's not about predicting the A. It's about what other things of the mechanism of splicing, in this example, has the model learned that we as biologists don't even know of. And that was impossible at the time of Irene's thesis, but now there are technologies out there or becoming available that are called explainable AI. Mostly comes out of the corner of medical prediction. We need to know why we trust the model. But I think as biologists, we can apply these approaches to learn stuff about biology that we currently don't know. Number one. Second one is 3D transcriptomics, 3D functional. This is a project instigated by Maurice that we currently, we got funding, that we currently pursue, and the idea is to build a 3D dimensional map of a tiny organ nose called the olfactory bulb that is required for our sense of smell, and it's reasonably small, and we want to have a 3D model at cellular resolution, not just being able to paint the cell in the right spot of the right type, but to have a description of the cell's functional competencies. What can this cell do? What can it communicate with from transcription profiles and lipidomics and proteomics? It's the technically most difficult thing I can currently imagine. But we're doing it, and Alex, a very talented PhD student, has just submitted her thesis. Wahapu, come on. Come up with an examination date so Alex can get on with life. So this is something we're pursuing. So the transformational technology here is 3D transcriptomics, called spatial. And the last one that is really close to my heart is the autism research framework. The work that I've described in Minds for Minds, instigated by Jesse, blame it all on you, and aiming for explaining to New Zealand families the cause and the reason and opportunities of their neurodevelopmental condition. And neurodevelopmental conditions are frequent. Individually, they are rare, but was it 17%? 17% of New Zealand families are affected by neurodevelopmental conditions. It doesn't include Alzheimer's or Huntington's. So the work that we've done is on a research level in Minds for Minds, we have last year, end of last year, transformed into a research clinic under an umbrella of the CDR up the hill. And Curtis, thanks to startup funding by the Freemason Society, and really our vision is to make this into a clinic where we don't just sequence, where we have research that can demonstrate the causality of variants that are currently not useful for clinicians because they don't have enough evidence. There's not enough knowledge attended to it. And the way we want to do it, and if you get this done in reasonable time, this will be a world-leading research clinic in the area of neurodevelopmental conditions. This is really something that we can aim for and can achieve. And believe it or not, we may be busy, but thank you for your patience. Thank you for your patience.